Thank you very much for the introduction. So today I would like to present our work on the effects of low magnitude, high frequency vibration, whether this is beneficial or detrimental. Our investigations were performed in a degenerative bovine disc organ culture model. We have no interests to declare. So our question comes from, as we all know, that disc degeneration associated with back pain has been described to be more prevalent in female adults in the ages ranging from 60 to 80. And that estrogen or the estrogen deficiency may be a contributing factor for disc degeneration in postmenopausal females. In this context, low magnitude, high frequency vibration has been increasingly used for the treatment of postmenopausal osteoporosis, using, for example, these vibration platforms that you can see on the image. This, however, vibration has been showing some conflicting results in the literature. A study performed on patients which were undergoing extended bed rest showed that this had a positive effect, so there was an attenuation of the detrimental changes of the morphological changes of the disc during the bed rest. However, in mice, it has been shown that exposure to low magnitude, high frequency vibration does have a detrimental effect on morphological disc changes. Therefore, our study went to focus on the effects of estrogen and on low magnitude, high frequency vibration on a particular degenerative bovine disc model. So experiments were done ex vivo. We wanted to know whether there is an effect of estrogen when it comes to disc matrix remodeling, production, and also whether this vibration has a different role when we have estrogen sufficiency or deficiency. For that, we have used healthy bovine tail discs, which we had injected with papain, so with a degrading enzyme that was used to simulate a degenerative environment, of course, up to a certain extent. 2 days later, we started applying the treatment, so either estrogen alone and or in combination with vibration. 2 days later, we looked at the gene expression of the cells. I will not show this data today, but 2 weeks later, we performed histological evaluations and biochemical evaluations of the treated discs. And I would like to start by showing you the data for the DNA content of these discs, as well as for the glycosaminoglycans, SCAGs, and for collagen. You can see on the left-hand side the data for the annulus fibrosus, AF, and on the right-hand side of each graph, the data for the nucleus pulposus, or NP. So regarding DNA, we could see that the application of vibration or estrogen combined with vibration after the treatment with the papain significantly increased the DNA content of the discs compared to both control and the papain treatment alone. So there was an increase of cell viability when these discs were vibration. Nevertheless, all treatments except vibration alone significantly decreased the GAG content. So the papain treatment per se decreased the GAG content, but not as much in the vibration group as in the others. Regarding collagen, we can appreciate that the group which was treated in combination of estrogen and vibration, significantly increased, on the other hand, the collagen content both in the annulus but also in the nucleus. Then we also went on to look at the production and distribution of pro-inflammatory enzymes. For example, I show here the results for interleukin-6 and of matrix-degrading enzymes that you can see on the right-hand side, for example, the example for MMP3. And here we could observe that all groups which were treated with the papain increased the production of these two factors. Additionally, the group which was treated with the estrogen in combination with the vibration increased IL-6 in the annulus and MMP3 in the annulus and the nucleus compared to papain treatment. Lastly, we also wanted to see whether this chemical content changes also translated in some morphological changes or alterations on the tissue, and we measured stiffness and strength of the annulus fibrosus tissue. And you can appreciate on both graphs that papain significantly increased the stiffness and the strength of the AF tissues compared to control. However, when we looked just at the peel stiffness, all the treatments that followed were able to decrease the stiffness, or these tissues presented lower stiffness in comparison to the papain-treated ones. Therefore, in combination, in summary, I would like that you go home with this take-home message, which is that papain digestion does induce disc degeneration. There are already some published models in a different context. That estrogen does not really seem to have an effect on protective against degeneration in this model. However, low magnitude, high frequency vibration does further accelerate the degeneration of these tissues. So in conclusion, low magnitude, high frequency vibration does seem to be detrimental in this degenerative bovine disc organ culture model. So with that, I would like to thank your attention, and I would like to call your interest if you're interested in our further research in this topic. We have also performed pressure measurements inside this disc, so go and please visit poster 411. Thank you.